Hi and welcome everyone. My name is Teresa Rummel. I'm a PhD student in the AirHead lab, and today I'm going to present you our new R package grandR for the analysis of nucleotide conversion RNA-seq data. First of all, let me introduce you to nucleotide conversion RNA-seq. Here specifically I'm going to talk about SLAM-seq, but there are similar approaches though, like TAC-seq or TALNAC-seq, that can also be analyzed with grandR. In SLAM-seq, cells are treated with a nucleoside analog, for example 4-SU, which then gets incorporated into newly synthesized RNA. Before sequencing, cells get treated with a chemical agent to convert 4-SU into cytosine or cytosine analog. Labeled, so newly synthesized, and unlabeled, so old RNA, can then be distinguished based on TTC mismatches that we can also identify in the reads. Unfortunately, concentrations that are well tolerated by cells commonly only replace 1 in 40 readings, resulting in a big percentage of reads that does not contain any mismatch, even though we have newly synthesized RNA. Luckily, our lab previously proposed a statistical solution to quantify labeled and unlabeled RNA without bias. By using a binomial mixture model, our grandSLAM approach provides unbiased new to total ratios, so the MTR, and also its posterior distribution. This can be taken further to downstream analysis. So what's the benefit of nucleotide conversion RNA-seq? Let's suppose we have an experiment with 3 replicates with and 3 replicates without perturbation. Without metabolic labeling, we get limited information. In gene 1 and 2, we don't see any change. In gene 3, there might be a slight upregulation, and in gene 4 and 5, we see a higher expression. Looking now at metabolic labeling data, we get way more information. In gene 1 and 2, we can estimate the half-lives. In gene 3, we see that there has indeed been an upregulation. In gene 4 and 5, we can tell why there is a change in gene expression. So taken together, this is a powerful tool to measure temporal dynamics of gene regulation. But the analysis of this data is challenging, and so far tools for this were lacking. That's why we developed grandR. grandR is a big toolkit that offers functions to do each step of the analysis, so up from quality control to publication-ready figures. If you know the Seurat package for single-cell data analysis, then you know where we got our inspiration from. So let's have a look at some of our tools. Since high concentrations of 4-SU or labeling of extended periods of time can affect cell viability or RNA metabolism, no quality control measurements for this type of data are needed. To control for such effects, in grandR we compare 4-SU labeled samples with equivalent 4-SU naive control samples. To showcase this, we analyzed here a dataset in which mouse embryonic stem cells were treated with 4-SU for 24 hours in a pulse chase experimental setup in order to estimate RNA half-lives. Quality control in grandR revealed that in a PCA, the no 4-SU and 4-SU treated samples segregated. By having a closer look, we saw that 447 out of roughly 7,000 genes were significantly and strongly regulated due to the long time 4-SU treatment. Kinetics can also be analyzed without chase by monitoring the drop of RNA over time. Such progressive labeling experiments provide accurate estimates when time points are chosen roughly in the range of the actual RNA half-life. Here we looked at an experiment where they did progressive labeling at 0, 2, 4, and 8 hours. Quality control here revealed that after 8 hours, but not before, the log-fold change of 4-SU to no 4-SU was correlated with the NTR rank. This is the drop in the right-hand plot, whereas on the left-hand plot we don't see such effects. In general, 4-SU concentrations should be chosen such that 4-SU-induced effects on RNA metabolism are minimized, also for later time points. With this quality control steps, we can assess such effects and also exclude samples from further analysis. In general, 4-SU labeling requires additional quality control steps, and we believe that analyzing this correlation here should be done routinely for such experiments. In order to estimate now kinetics from such type of data, we need a model. The commonly used model is this, in which a gene is produced with a fixed synthesis rate and destroyed with a fixed degradation rate. Consequently, the number of RNA molecules will reach a steady-state level. The speed by which non-steady-state levels approach steady state can be characterized by the half-life, which would be here the intersection point. In the past, different variants of this model have been used to estimate kinetic parameters. We implemented 4 different models in grandR: linear regression, nonlinear least square regression, pools R, which is an R package which was made for slightly different data, and our own approach using the NTR from GrandSlam. In order to compare all of these methods, we simulated progressive labeling, nucleotide conversion seq data. The simulation is also implemented in grandR. And then we checked if the half-lives correlate well with the truth. And in general, this is the case for all methods, but NNLS and NTR correlated the best. Another important thing to consider is that FORCE is not immediately available for transcription once the cells are cultured on the FORCE media. It first needs to be transported and also processed. So the concentrations of active FORCE increase until saturation, and before, so in early time points, the samples contain less FORCE than later. So let's, for example, say we have the 1 hour time point. But in the first 30 minutes, their concentrations were so long that effective labeling only started after that. So effective labeling time would be half an hour instead of 1 hour. And we can actually observe this effect in data. So here in the line plot, we can see that the 1 hour time point would fit way better around 30 minutes. And this is not only true for this one gene, but it's a global effect. If we look here at the box plot, we see that the relative residual was negative in the early time points for almost all genes. But with grandR, we introduced a recalibration for this using a joint maximum likelihood. And we see that after calibration, this effect can be completely rescued. And this is especially critical when we want to compare samples with distinct labeling times, and of course it also is important for estimating degradation and synthesis rates. But there are also applications besides from progressive labeling data. Like I told you in the introduction, data from a single snapshot is already more sensitive to detect short-term changes of gene expression upon stimuli than without FORCE. So we developed a statistical model to estimate degradation and synthesis rates from this type of data. To test our approach, we simulated data for 2 conditions, with 2 hours labeling. One condition is steady state, the other is either perturbed synthesis or perturbed half-life or unperturbed control. Previously, to infer anything about synthesis or stability, differential expression analysis has been applied to old and new RNA. If we look at the log-fold change of new RNA, this also correlates very well with the log-fold change of the synthesis. But the log-fold change of the old RNA does not correlate with the log-fold change of the half-life. It's also important to consider that even though in the early time points here only the half-life were perturbed, we can see massive changes in new RNA that with this approach could be mistaken for changes in synthesis. So we wanted to improve this and get actual synthesis and half-life estimates using a Bayesian approach. If we look at our maximum a posteriori values, we see that they were unbiased for synthesis and actually even more accurate than log-fold changes of new RNA. And also for half-life, they were more accurate. Next, we used the rope analysis, so regions of practical equivalence, to quantify significant changes of degradation or synthesis with our approach. We compared it with DSIC2 for new and old RNA. So in these line plots, we see the bins of the genes over the log-fold change of synthesis or half-life. The lines show the percentage of genes in a bin with detected regulation. So first we looked at the simulated data with perturbed synthesis. Here we see that ROPE and DSIC2 showed similar sensitivity and specificity. So both only detected changes in synthesis. So here the yellow line. Next, we focused at simulated data with perturbed half-lives. Here on the right-hand plot, we saw that our model, so the rope, only detected changes in half-life, which is the blue solid line, whereas the ECCB detected changes in half-life in both, so the blue and red dashed line. In general, our approach can accurately differentiate between changes in synthesis and degradation and is more sensitive than analyzing the old RNA for detecting changes in half-life. Finally, we applied our differential gene expression modeling approach to a published dataset. Here we have cells after decrin-mediated depletion of BAMP. We had samples from multiple time points, so 1, 2, 4, 6, and 20 hours after depletion, and all of them were labeled with 4U prior to sequencing. Next, we estimated RNA synthesis log4 changes with our Bayesian approach for each time point. This enabled us to compare all time points even though they have different labeling times. For each time point, we saw that the RNA synthesis log4 changes for BAMP targets were shifted towards negative values compared to non-targets. This is remarkable, especially for the 1 hour time point where force labeling only was 30 minutes and shows that the synthesis rates were reduced immediately once BAMP was depleted from the cells and stayed constant for at least 20 hours. To further investigate this, we analyzed the synthesis LFC posterior distributions for individual genes, and we saw substantial gene-specific differences. Some BAMP targets like TEF1D showing gradually decreasing synthesis rates with effective downregulation only later than the 1 hour time point. For others like HERC1 or TOP-GCP5, we saw that synthesis rates dropped early and rose again later, suggesting negative feedback loops. So in conclusion, the model implemented in grandR can be used to uncover detailed information about gene regulation from snapshot experiments and thereby reflecting general trends and is able to generate testable hypotheses of individual genes. grandR also offers many functions for visualization that can be either accessed programmatically or can be loaded into a Shiny-based web interface. This web interface mainly consists of 2 tabs. First is the gene level, the second is the global level. On the gene level, we have our analysis table, which we can search and filter. If we select a gene here, gene level plots will appear for this gene. Those plots are completely customizable and interchangeable. In this first tab, we also have the opportunity to download the data or the table. In the global level, we can load several global plots. In those global plots, we can select genes that will then appear in the right-hand box. We can set a highlight, and the highlighted genes will then be read in all global plots. Also, we can use genes identified here, of course, then on the single gene level and vice versa. In general, this is a really handy tool for collaborating with other labs, sharing data, and just for intuitive data analysis. So for all of you who now want to use grandR, it's publicly available and we have extensive documentation on our lab page, including nice vignettes and a cheat sheet. Also, our paper is now published, so feel free to go ahead and read it. And with this, I want to thank everyone involved in this project, especially Florian and Gregory, and you for your attention.